I'm Madeline, and I will be presenting this article on the mechanism of helicase loading as studied by single-molecule methods. So we discussed MCM 2-7 and origin licensing in class with Article 2 shown on the left, but that paper from 2009 only revealed the structure of the double hexamer and its requirements for loading onto DNA. The text that I highlighted from Article 2 is from the discussion section and suggests an area of future study. The article that I will be discussing now is from 2015, and it's one such example of future work on elucidating the mechanism of MCM 2-7 loading. The single molecule assays in this paper used co-localized fluorescently tagged origin DNA and individual associated proteins. On the left is the schematic of the assay where origin-containing DNA, labeled with Alexa Fluor 488, was immobilized on microscope slides. Each experiment also tagged one or two of the associated proteins, and this schematic represents the first experiment where the MCM 2-7 hexamer was visualized by SNAP tagged MCM4 labeled with 549 fluorophore. Each tagged molecule was studied in real time with total internal reflection microscopy as shown on the right. The red arrows indicate representative co-localization pairs of DNA and MCM 2-7. The benefit of single molecule methods is that individual origin complexes can be simultaneously studied in real time without being averaged. So for the first experiment, panel B shows the increase in intensity of MCM 2-7 fluorescence for two DNA molecules over time. There are two distinct intensities indicated by the red and blue arrows. The stepwise fashion suggests that each MCM 2-7 is recruited sequentially, where the red arrow represents recruitment of the first MCM hexamer and the blue arrow represents recruitment of the second hexamer. Panel C shows the dwell times of MCM 2-7 on DNA, and the main takeaway from the histogram and inset is that the distribution is multi-exponential, which suggests at least two distinct types of MCM 2-7 association with DNA. Going back to panel B, to eliminate the possibility that the steps can be explained by simultaneous recruitment of already formed dimers, the researchers used photobleaching to count the number of MCM hexamers associated with the DNA. The tethered DNA was washed with buffer to remove non-DNA associated MCM 2-7, and then fluorescence increase was compared to the loss of fluorescence. The key point is that photobleaching occurs one molecule at a time. The top panel of D shows these results, with the most important data being that no two-step photobleaching was associated with a one-step up in fluorescence. Panel E and the bottom of panel D are a repeat of the photobleaching experiment with a high salt wash to remove any unloaded MCM 2-7 complexes. The combined conclusion of these experiments is that recruitment and loading of MCM 2-7 to DNA occurs at a one-at-a-time sequence. The next experiments examine the loading of CDT1 in relation to the timeline of MCM 2-7 association. Panel A is similar to figure 1b, but this time co-localized MCM 2-7 and CDT1 are shown for a single origin. The baseline for MCM 2-7 is shifted up relative to CDT1, and we can see from the fluorescence in the plot and the single frame micrographs that MCM 2-7 and CDT1 association with DNA are concurrent. Also, distinct CDT1 molecules are associated with each MCM 2-7 loading. The green arrow indicates simultaneous release of MCM 2-7 and CDT1, while the black arrows indicate release of CDT1 without release of MCM 2-7. Panel B again shows a multi-exponential distribution of dwell times that suggests at least two types of CDT1 association with DNA. Accordingly, panel C and D show two types of observed MCM 2-7 and CDT1 releases. CDT1 and MCM 2-7 were either released simultaneously with relatively short CDT1 dwell times, or CDT1 was released before MCM 2-7, in which case the dwell times were more varied and longer on average. The experiments from figure 2 were repeated with CDC6, and the fluorescence intensity record shown in panel A reveals distinct CDC6 molecules for each MCM 2-7 association. The record of micrographs show that CDC6 arrives at the DNA origin before MCM 2-7. The sequential loading of CDC6 and MCM 2-7 for both the first and second MCM 2-7 was confirmed with the histograms in panel B. The blue bars, zoomed in here on the bottom, represent the difference between the time of MCM 2-7 arrival and the closest time of CDC6 arrival on the same DNA molecule. The majority of time differences being greater than 0 confirms sequential arrival. The red bars are a randomized control for the MCM 2 through 7 and CDC6 on different molecules to prove that the arrival time difference is not coincidental. The next question was the order of CDT1 and CDC6 loading and release. Panel A contains 3 representative fluorescence intensity records that show CDC6 arrives before CDT1 and releases first. Panel B is a plot of CDC6 release time on the x-axis versus CDT1 release time on the y-axis, both measured from the time when both proteins are present. The red line represents where the data points would fall if release time was simultaneous, but CDC6 release occurred first in 95.5% of the time measurements. To put these results back in context of double hexamer formation, release times for CDC6 and CDT1 were plotted in relation to MCM 2 through 7 recruitment in Panel C. Panels D and E separate the CDC6 and CDT1 data for first and second MCM 2 through 7 arrival, revealing slower release of both proteins on the second MCM 2 through 7 loading event. Another Cosmos experiment was done with labeled MCM 2 through 7 and ORC to determine how many ORC molecules are associated with MCM 2 through 7 loading and when. The intensity record shown reveals that a single ORC molecule associates with DNA in MCM 2 through 7 and remains present for the first and second MCM 2 through 7 recruitment. Panel B shows an examination of the expected number of ORC molecules based on models described in supplemental information compared to what was experimentally observed. The results revealed that only one ORC molecule is present for the recruitment of 2 MCM 2 through 7 hexamers, consistent with model 1 or 4. Panels C and D are more time plots, and they reveal that ORC is released quickly after the second MCM 2 through 7 recruitment, and that there is no significant difference between ORC and CDT1 release times. FRET was used in the last main experiment to investigate the recruitment of the second MCM 2 through 7 and formation of the double hexamer. The MCM 7 subunits were labeled with donor and acceptor fluorophores in an equimolar ratio, and excitation wavelengths were alternated to distinguish arrival of each MCM 2 through 7, as well as FRET. Representative fluorescence records for 2 origins are shown in Panel B, and the bottom panel shows the calculated FRET efficiency associated with double hexamer formation. The histogram of FRET efficiency on the left shows the presence of both labeled hexamers with blue bars and only the donor labeled hexamer with empty gray bars. The high FRET efficiency here, when 2 hexamers are present, corresponds to the double hexamer formation following recruitment of a second MCM 2 through 7 hexamer. Panel D puts double hexamer formation on a time scale, and the authors found that double hexamer formation, which is represented by the green FRET survival curve, precedes the release of CDC6 and CDT1, indicating that they are independent events. Finally, we have the results of each experiment summarized in a schematic of the proposed mechanism for helicase loading as related to loading of the MCM 2 through 7 double hexamer. The ORC and CDC6 are recruited first, and then the first MCM 2 through 7 and CDT1 are recruited simultaneously. CDC6 and CDT1 release after the first recruitment of the first MCM 2 through 7 hexamer, while the ORC remains. And then another CDC6 molecule is recruited, followed by another simultaneous association of MCM 2 through 7 and CDT1. Once the double hexamer is formed, the CDT1 molecule is released, followed by concurrent release of CDT1 and the ORC. From there, helicase loading onto the MCM 2 through 7 double hexamer occurs.